Imagine standing at the edge of the ocean. Not today's ocean. Not the blue, familiar world of sharks and whales and sunlit coral reefs. This is a different ocean, an ancient one. An ocean that existed so long ago that the very continents beneath it had not yet taken the shapes we know. The air above the surface carries more oxygen than you're used to. The water itself is warmer, shallower, spreading across vast inland seas that cover what will one day become North America, Europe, Morocco, China. It is 380 million years ago, and something is moving in the deep. You can't see it yet, but you can feel it. A pressure change in the water, a vibration that moves through the seafloor and up through your bones. Something large is approaching, something that does not need to hurry, because nothing in this ocean has ever given it a reason to. Then it rises, and the first thing you notice is not the size, though the size is staggering. The first thing you notice is the head, because the head of this animal looks less like a living creature and more like a siege weapon. Flat, armored, encased from the tip of the snout to the back of the skull in interlocking plates of solid bone, layered over each other like the hull of a warship. No soft tissue exposed, no weakness visible. Just bone and mass and forward momentum. This is Dunkleosteus terrelli. It is not a shark. It is not a bony fish in any sense you would recognize today. It belongs to a group of animals so ancient, so alien in their construction, that modern biology had to invent an entirely separate category just to contain them. It is an arthrodire placoderm, an armored fish from a lineage that has been completely, utterly erased from the planet. Every single relative it ever had is gone. The entire class wiped out. What you are looking at is not just an extinct animal.
It is a ghost from a branch of life that reached an evolutionary dead end so total that it left almost nothing behind except bone. And yet, the bone it left behind tells one of the most extraordinary stories in the history of life on Earth. Dunkleosteus did not have teeth. Let that settle for a moment. The most powerful bite ever measured in a fish, a bite force that, according to biomechanical modeling published in the journal Proceedings of the Royal Society, exceeded 5,000 newtons at the tip of its jaws, and it achieved that force without a single tooth in its head. Instead, it had blades, self-sharpening blades of exposed bone grown directly from the jaw structure itself that worked against each other like a pair of scissors the size of a dining table. Every time those jaws closed, the edges honed themselves sharper. The more it fed, the more dangerous it became. It could open those jaws in approximately 1/50th of a second. To put that in context, a human blink takes roughly 1/3 of a second. By the time your eyes have closed and opened again, Dunkleosteus has already bitten down, released, and reset. At full size, this animal stretched somewhere between 5 and 6 meters in length, roughly the size of a large pickup truck, though some estimates based on fragmentary evidence from the largest known specimens push that figure toward 8 or possibly 10 meters. Its front half, encased in bone armor, may have weighed over a metric ton on its own. The rear half of the body, by contrast, was soft and unarmored, almost certainly flexible, muscular, built for sudden bursts of acceleration rather than sustained pursuit. It was an ambush predator in a world that had never seen anything like it. And for roughly 40 million years, it ruled. 40 million years. Consider that number. Modern humans have existed for roughly 300,000 years. We consider our civilization ancient when it stretches back 5,000 years. Dunkleosteus patrolled these primordial seas for a span of time so immense that if you compressed it into a single day, our entire human history would not register as a single heartbeat. In that time, it did not merely survive, it dominated. It shaped the evolution of every other creature in its ecosystem. Fish developed new escape strategies because of Dunkleosteus. Prey species evolved armoring of their own. Entire lineages bent themselves around the existence of this one animal, this armored predator at the absolute top of the Devonian food chain. And then, at the end of the Devonian period, approximately 359 million years ago, it vanished. Not slowly, not gradually, but in one of the most devastating mass extinction events in the history of our planet. An event so catastrophic that it erased more than 70% of all species on Earth and nearly extinguished the vertebrate lineage entirely. Nearly. Because the story of Dunkleosteus is not just the story of a monster, it is the story of what came before us, of the ocean that built the template for every predator that would ever follow, of an evolutionary experiment so successful, so terrifyingly effective, that the universe allowed it to run for 40 million years before finally pulling the curtain down. This is that story, and it begins as all the best stories do, not with a triumph, but with a discovery. A single bone, a fragment of armor, pulled from a cliff face in Ohio by a man who had no idea what he was holding. The year is 1867. The place is a small town called Sheffield Lake, on the southern shore of Lake Erie in the state of Ohio. A man named J. Terrell is working in a shale quarry. This is not glamorous work. It is slow, physical, repetitive. The kind of labor that leaves your hands cracked and your back aching before noon. The quarry sits inside a formation of dark, compacted shale that geologists would later classify as part of the Cleveland Shale member, a deposit laid down during the late Devonian period in the sediment of an ancient, shallow sea that once stretched across the interior of North America. J. Terrell is not a paleontologist. He is not looking for fossils. He is cutting rock. But the rock gives him something anyway. He finds a piece of bone. Not a small piece. A large, dense, heavily structured fragment of what is clearly a skull. The bone is unlike anything he has seen before. It is thick, layered, almost architectural in its construction, like something engineered rather than grown. He sets it aside. He finds more pieces nearby. He collects them because even a man with no scientific training can recognize when he is holding something that does not belong to any animal he has ever encountered. He brings the fragments to a local physician and amateur naturalist named John Strong Newberry. Newberry looks at the bones, and even Newberry, who has studied fossils for years, is not entirely sure what he is looking at. The structure is wrong for any known fish. Too heavy, too armored, too ancient in its geometry. He publishes a preliminary description, names the specimen, and sends it into the scientific community with a note that amounts to, "This is something we have not seen before." He is right, but he does not yet understand how right he is. The name Dunkleosteus terrelli comes later, a tribute to two men. The species name, terrelli, honors J. Terrell, the quarry worker who first pulled the bones from the rock. The genus name, Dunkleosteus, honors David Dunkel, a paleontologist at the Cleveland Museum of Natural History who spent decades studying these animals and who, more than perhaps any other single researcher in the 19th and 20th centuries, shaped our early understanding of what this creature was. The name itself, in Greek, means Dunkel's bone.
a simple name for a complex animal. For decades after Newberry's initial description, the scientific understanding of Dunkleosteus advanced slowly. The Cleveland Shale continued to yield specimens, not complete skeletons but fragments, skull plates, jaw sections, portions of the thoracic armor that encased the front of the body. Because the rear half of Dunkleosteus was made of soft tissue rather than bone, it almost never fossilized. The ocean floor consumed it too quickly. What survived was what was hard: the armor, the skull, the jaw. And so for a long time, that was all paleontologists had to work with. This created a fundamental problem that haunts Dunkleosteus research to this day. We do not have a complete specimen. Not one. Not a single individual of Dunkleosteus terrelli has ever been found with both its armored front half and its soft rear half preserved together. Every size estimate, every reconstruction, every biomechanical model ever produced for this animal is based on extrapolation. Researchers take the dimensions of the skull and thoracic shield, compare them to more completely preserved relatives within the placoderm family, apply scaling ratios, and work backward to produce a body estimate. And depending on which scaling method you use and which relatives you compare it to, you get very different animals. The conservative estimates place Dunkleosteus terrelli at around 5 to 6 meters in length. These models compare the skull dimensions to well-preserved smaller placoderms and scale proportionally. The result is a formidable but not extraordinary predator. Large, yes, powerful, certainly, but within the range of other apex predators we know from the fossil record. Then there are the other estimates. Some researchers, looking at the largest known skull fragments and applying different scaling relationships, argue for animals approaching 8, 9, even 10 meters in length. A 10-meter Dunkleosteus would have been comparable in size to a large great white shark. Its skull alone, at full scale, would have been over 1 meter long and nearly as wide. The thoracic armor covering its chest and shoulders would have weighed several hundred kilograms on its own. The scientific community has not reached consensus. The debate is ongoing, and the honest answer, as of our current state of knowledge, is that we do not know exactly how large the biggest individuals grew, because we have never found one complete. What we do know is what the Cleveland Shale has given us, and what it has given us is extraordinary. The Cleveland Museum of Natural History holds some of the finest Dunkleosteus material in the world. Their mounted reconstruction, assembled from multiple specimens, composite in nature, scientifically reviewed and revised multiple times over the decades, gives visitors a physical sense of the animal's scale and architecture that no illustration can fully convey. Standing in front of that skull, looking at the interlocking plates of cranial armor, at the exposed bone blades of the jaw, at the orbital cavities where the eyes once sat, you feel something that goes beyond intellectual appreciation. You feel old. You feel small. Because the animal looking back at you, or rather, the ghost of the animal looking back at you, is from a world so ancient that our entire evolutionary lineage, from the first primitive mammal-like reptile to you standing in that museum, would fit inside its lifetime with room to spare. But here is the detail that first drew serious scientific attention beyond the initial discovery. In 1873, workers in the same Cleveland Shale deposits began finding not just individual bone plates, but clusters, multiple specimens in close proximity, overlapping armor fragments from several different individuals, preserved together in the same sedimentary layer. This was unusual. It suggested something about the behavior of these animals or about the conditions at the time of their death
that a single isolated specimen could not reveal. Were they schooling? Were they territorial and dying in disputes? Were they caught in some environmental event, a sudden drop in oxygen, a toxic upwelling, a catastrophic storm that killed multiple individuals at once and deposited them in the same place? Nobody knew then, and nobody knows with certainty now. But those clustered deposits gave paleontologists something invaluable. They gave them sample sizes. They gave them multiple individuals of different ages and sizes. They allowed researchers to begin building a picture not just of one animal, but of a population, of a species, of a creature that lived and died and aged and competed in a real ecosystem over a real span of geological time. And one of the things those multiple specimens revealed was this: Dunkleosteus was not alone. The late Devonian seas that it called home were not a simple environment with one dominant predator and everything else cowering beneath it. They were complex, layered, populated by a staggering diversity of placoderms, early sharks, primitive bony fishes, and invertebrates that we are still in the process of cataloging and understanding. Dunkleosteus was the apex, but it was an apex embedded in a web of ecological relationships that stretched in every direction. To understand what Dunkleosteus really was, what it ate, how it hunted, how it interacted with its world, you have to understand that world first. And that world was stranger than anything alive today. Close your eyes for a moment and try to picture the Earth as it existed 380 million years ago. Not as a barren rock, not as a lifeless void. The planet is very much alive, but alive in a way that would feel profoundly alien to any modern observer. The oxygen content of the atmosphere is slightly lower than today, but the carbon dioxide levels are dramatically higher, producing a greenhouse effect that keeps global temperatures elevated, warm even at the poles, sweltering near the equator. There are no ice caps. There is no permanent glaciation anywhere on the planet. The world is wet and green and ancient in a way that goes beyond description. This is the Devonian period. It runs from approximately 419 million years ago to 359 million years ago. 60 million years of geological time divided by geologists into 3 stages: early, middle, and late. Dunkleosteus terrelli belongs to the late Devonian, specifically the Frasnian and Fominian stages, the final chapters of this long period before the catastrophe that ends it. The Devonian has a nickname among paleontologists. They call it the Age of Fishes. And that name, simple as it sounds, does not come close to capturing what the oceans of this period actually were. Because the Devonian seas were not merely full of fish. They were the crucible in which fish, vertebrate life in water, reached its first great evolutionary flowering. Every major body plan that vertebrates would ever develop, every fundamental innovation in jaw structure, in locomotion, in sensory systems, in reproduction, most of it was being invented and tested and refined in the oceans of the Devonian. What we think of today as the basic template of a fish, paired fins, a backbone, a skull with movable jaws, that template was still new, still being experimented with, still being pushed in directions that evolution would ultimately abandon. And the continents bearing witness to all of this look nothing like the world we know. In the late Devonian, the supercontinent Gondwana dominates the southern hemisphere, a vast landmass that contains what will eventually become South America, Africa, Antarctica, Australia, and India, all fused together in a single continent of almost incomprehensible size. To the north, the smaller continents of Laurussia, a merger of what will become North America, Europe, and parts of Asia, sit straddling the equator, low-lying and frequently flooded by shallow epicontinental seas. These epicontinental seas are critical to understanding Dunkleosteus. The western interior seaway, the ancient ocean that covered much of what is now the American Midwest, is not yet the vast body of water it will become during the Cretaceous. But its Devonian predecessor, sometimes called the Appalachian Basin or the Cleveland Sea, is already substantial. It is warm. It is shallow. In many areas no deeper than a few hundred meters. Its floor is rich in organic sediment, layered in the fine dark shale that will eventually compress and harden into the rock formations from which Dunkleosteus will be exhumed hundreds of millions of years later. This sea is stratified. The upper layers are oxygenated, alive, productive. The deeper layers, beneath the thermocline, are anoxic, starved of oxygen, toxic to most life, a dead zone that paradoxically becomes a preservation machine. When animals die in the upper layers and sink, they pass through the oxygenated zone where decomposition and scavengers would normally destroy them, and settle into the anoxic sediment below, where bacteria cannot thrive and decomposition slows to almost nothing. This is why we have Dunkleosteus at all. The same conditions that made the Cleveland Sea a productive hunting ground for an apex predator also made it, after death, the perfect burial chamber for that predator's remains. The ocean that sustained Dunkleosteus in life became the archive that preserved it in death. There is something almost deliberate about that, a neatness that nature, of course, had no intention of providing. Now picture the coastline of this ancient sea. The land bordering the Devonian seas is undergoing its own revolution.
For most of Earth's history up to this point, the land has been largely bare, populated by microbial mats, early mosses, primitive plants that cling to the edges of water but have not yet conquered the interior. But during the Devonian, something extraordinary is happening. Plants are exploding, truly exploding, diversifying, growing taller, developing woody tissue for the first time, pushing inland, climbing hillsides, reaching toward the elevated light. By the late Devonian, the first genuine forests are appearing on Earth. Trees, real trees with trunks and root systems and canopies, are growing along the margins of the continents for the first time in the planet's 4.5 billion year history. Archaeopteris, a tree that can reach heights of 30 meters, spreads across the Devonian landscape in dense stands that would not look entirely out of place in a modern forest, if you could ignore the absence of birdsong, the silence of a world without insects capable of flight, the strange quality of air that carries no pollen you would recognize. These forests have a profound effect on the ocean. As trees develop deep root systems, they begin breaking apart rock at unprecedented rates. Minerals, phosphorus, calcium, iron, are released into rivers and carried down to the sea. This nutrient influx triggers massive algal blooms in the shallow coastal waters. The blooms die and sink. Bacteria consuming the dead algae strip oxygen from the water. Dead zones expand. The same anoxic conditions that will later preserve Dunkleosteus fossils are being created and amplified, in part, by the growth of forests on land. Everything is connected. Everything in this ancient world is entangled with everything else in ways that took geologists and ecologists decades to untangle. And swimming through the middle of all of this, through the warm, stratified waters of the Cleveland Sea,
through the nutrient-rich coastal shallows, through a world in the process of being remade by the march of forests across the continents, is an ecosystem of staggering complexity. The base of the food web is built on phytoplankton and algae, just as it is today. Above that, vast populations of small invertebrates, primitive crustaceans, early nautiloids, the coiled ancestors of modern cephalopods, filter and graze and are in turn consumed by small fish. These small fish include some of the earliest known sharks, not the sleek torpedo-shaped animals we associate with that word today, but primitive experimental forms like Cladocellaceae, a meter-long predator with a deeply forked tail and no calcified skeleton, whose cartilaginous remains are preserved in stunning detail in the same Cleveland shale deposits that yield Dunkleosteus. Cladocellaceae is fast, more streamlined than almost anything else in the Devonian Sea. Its tail design suggests it was built for speed, a pursuit predator that chased small fish through open water. In some specimens preserved in the Cleveland Shale, scientists have found the remains of other fish inside the body cavity, swallowed whole and tail first, suggesting Cladocellaceae caught its prey from behind at speed and consumed it in a single fluid motion. It is not at the top of the food chain, not even close. Above Cladocellaceae, above the smaller placoderms, above the armored, jawless fish still lingering from earlier periods, above all of them is Dunkleosteus. But Dunkleosteus is not the only large predator in these waters. And this is where the story becomes more interesting, more layered, more true to the messy reality of how ecosystems actually function.
Alongside Dunkleosteus in the late Devonian seas, there are other large arthrodires, other armored predators of the same fundamental body plan, competing for the same resources, occupying overlapping ecological niches. Titanichthys, for example, is nearly as large as Dunkleosteus. Some estimates place it in the same size range, but its jaw structure is fundamentally different. Where Dunkleosteus has those devastating bone blades designed for cutting and crushing, Titanichthys has reduced, blunt oral surfaces that many researchers now believe were adapted for filter feeding. A giant in the same waters as Dunkleosteus, but eating at a completely different level of the food web. Not a competitor, almost an ecological mirror image. Same architecture, radically different strategy. Then there is Gorgonichthys, another large arthrodire, with a skull structure suggesting it occupied a predatory niche similar to Dunkleosteus, but perhaps specialized for different prey sizes or different hunting depths. And Eastmonosteus, and Hindsichthys. The late Devonian arthrodires are not a single species dominating a simple ecosystem. They are a radiation, a diverse group of related animals that have filled multiple ecological roles in the same ocean, the way that today's sharks occupy everything from filter feeding to apex predation to deep-sea scavenging. This diversity is important for understanding Dunkleosteus, because when we talk about Dunkleosteus as the apex predator of the Devonian, we are not describing a lonely king presiding over a simple court. We are describing an animal embedded in a complex, competitive, ecologically rich world. A world where resources were contested, where specialization was rewarded, where the evolutionary pressure to be better, faster, more effective was constant and relentless. And Dunkleosteus, in that world, was the answer to every question evolution had asked about what a large marine predator could be. The armor, the jaw, the speed. The sensory systems that are still, in many ways, poorly understood. All of it shaped by 40 million years of competition in an ocean that no longer exists. To understand how those weapons worked,
how that jaw actually functioned, what it was capable of, and what the science really says about the most powerful bite in fish history. We need to go deeper, much deeper. Start with the jaw, because the jaw of Dunkleosteus terrelli is not merely a weapon. It is an engineering solution, a piece of biological machinery so precisely constructed, so mechanically elegant, that when researchers at the University of Chicago first ran it through a full biomechanical analysis in 2006, the results stopped them cold. The study, led by Philip Anderson and Mark Westneat and published in the journal Proceedings of the Royal Society B, was the first rigorous quantitative examination of how the Dunkleosteus jaw actually functioned. Using computer modeling based on the dimensions of multiple well-preserved specimens, Anderson and Westneat reconstructed the muscle architecture, the lever mechanics, the joint geometry, everything that determines how a jaw opens, how fast it moves, and how much force it generates. What they found was not what they expected. The jaw of Dunkleosteus does not work the way the jaw of a modern predator works. In most living predators, sharks, crocodiles, large cats, the jaw opens primarily by dropping the lower mandible downward. The skull stays relatively fixed. The lower jaw descends, the mouth opens, the prey is caught. Simple. Effective. The design that evolution has returned to again and again across hundreds of millions of years. Dunkleosteus does something different. Its skull moves. The entire cranial unit, the massive armored head, the skull plates, the orbital region containing the eyes, rotates upward on a joint located at the back of the skull where it meets the thoracic shield covering the shoulder. At the same moment, the lower jaw drops. Both movements happen simultaneously, driven by a complex arrangement of muscles attached to the armor plates. The result is a jaw that opens not just downward, but in two directions at once, the upper jaw lifting away from the lower as the lower descends away from the upper. This mechanism, called craniokinesis combined with hyoid depression, creates a gape that is dramatically larger than either movement alone could produce. It also creates something else: a rapid drop in pressure inside the oral cavity as the volume expands so suddenly. This pressure drop generates suction, the same principle that makes a syringe draw fluid inward when you pull the plunger. As the jaws open at extraordinary speed, water and everything in it is drawn toward the expanding cavity between them. Prey does not just get bitten by Dunkleosteus, it gets inhaled. Anderson and Westneat calculated that the jaws could open completely in approximately 50 milliseconds. That 1/50th of a second mentioned earlier is not an approximation, but the result of direct mechanical modeling. At that speed, the suction effect would have been significant enough to draw prey inward even if the initial approach missed by a small margin. This would have made Dunkleosteus effective not just as a pursuit predator, but as an ambush specialist, a hunter that could afford to be slightly imprecise because the physics of its own jaw mechanics would correct for minor targeting errors. Then the jaws close. And this is where the numbers become almost difficult to process. Anderson and Westneat's model calculated a bite force at the tip of the jaw of approximately 5,000 newtons, roughly equivalent to the force exerted by 500 kilograms pressing straight down on a single point. At the rear of the jaw, where the blades meet in the deepest part of the bite, that force climbs to approximately 7,500 newtons. For context, the bite force of a large great white shark is estimated at approximately 18,000 newtons. But the great white is applying that force across multiple teeth distributed along a curved jaw. The force per point of contact is distributed, dispersed, spread. Dunkleosteus concentrates its force onto the two edges of exposed bone blade that meet first. The geometry of the bite means that force is applied at a single intersecting line, like scissors, rather than spread across a surface. The effective cutting pressure at that intersection point, according to the Anderson and Westneat model, would have been sufficient to shear through the bone armor of other placoderms. Not just bite through soft tissue, shear through bone. And those blades are self-sharpening. This detail deserves more attention than it typically receives in popular accounts of Dunkleosteus, because it represents something genuinely remarkable in the history of biological weaponry. The oral plates of Dunkleosteus, the exposed bone surfaces that form the cutting edges, wear against each other every time the jaws close. In most biological cutting structures, wear means degradation. A tooth that wears down becomes less effective. An edge that dulls loses its function. This is why sharks continuously replace their teeth throughout their lives, a conveyor belt of new cutting surfaces moving forward as old ones wear and fall away. Dunkleosteus cannot replace its oral plates. They are not teeth. They are part of the jaw structure itself. If they simply wore down, the animal would gradually lose its ability to feed and would die. But the geometry of the bite prevents this. The two opposing bone surfaces wear against each other at precisely the angle that maintains a sharp edge, like a self-stropping blade. The wear pattern itself becomes the sharpening mechanism. As the animal feeds, as the surfaces grind together, the angle is maintained, the edge is preserved, and the weapon becomes no less effective with use. Evolution, working across hundreds of millions of years of trial and error, produced a cutting system that improves with use. No human engineer designed anything comparable until the development of self-sharpening industrial blades in the 20th century. Now consider the armor. The head and thoracic shield of Dunkleosteus are composed of dermal bone, bone that grows within the skin itself rather than from an internal skeleton. This is an ancient system, common to many early vertebrates, but carried to its most extreme expression in the arthrodire placoderms. The plates of Dunkleosteus are not a single layer of bone. They are multiple layers, interlocked at their edges through a system of ridges and grooves that distribute stress across the entire shield rather than concentrating it at any single point. Think of it as biological composite armor. The outer surface is dense, hard, resistant to puncture and abrasion. Beneath it, the layering changes. Different densities, different orientations of bone fibers working together to absorb and redirect impact energy. When another predator or competitor struck the shield of Dunkleosteus, the force was not transmitted directly to the underlying skull. It was spread, dampened, dissipated across the entire structure. CT scanning of Dunkleosteus skull material, work carried out using high-resolution computed tomography at several institutions, including detailed analysis of specimens in the Cleveland Museum collection, has revealed internal structures in the bone that researchers are still working to fully interpret. Channels that may represent pathways for blood vessels or sensory nerves. Variations in bone density that suggest different regions of the shield were optimized for different mechanical functions. Some areas appear built to resist biting forces from the outside. Others seem structured to anchor the muscles responsible for driving the craniokinesis system. The armor is not just protection. It is also a structural framework for the entire feeding mechanism. Without the armor, the jaw cannot function. The two systems, protection and predation, are not separate adaptations bolted together by evolution. They are a single integrated system in which each element depends on the others. The armor provides the rigid anchoring points that the jaw muscles need to generate force. The jaw generates the force that makes the armor necessary. Remove one and the other loses its function. This is why the placoderm body plan, once established, became so stable, not because it was perfect, but because it was deeply integrated. Changing one part meant changing everything. But the jaw and the armor together still leave a fundamental question unanswered. How did Dunkleosteus actually hunt? We have the weapon, we have the defense, we have the biomechanics of the bite. But understanding how this animal behaved in life, how it located prey, how it approached, how it struck, how it consumed, requires looking beyond the skull plates and into the more fragmentary evidence of behavior preserved in the fossil record. And that evidence, when you find it, is visceral. In multiple locations within the Cleveland Shale and in equivalent late Devonian deposits in other parts of the world, researchers have found something distinctive associated with sites where Dunkleosteus remains are concentrated. They have found boluses, compressed masses of partially digested bone and organic material expelled by the animal after feeding. Similar to the pellets produced by modern owls and raptors, indigestible material compacted in the stomach and regurgitated. These boluses contain fragments of bone from prey animals, fish vertebrae, fragments of placoderm armor plate, the remains of sharks and smaller predators compressed and partially dissolved by stomach acid, then expelled as waste. By analyzing the contents of these boluses, researchers have been able to construct a partial list of what Dunkleosteus was eating, and the list is not limited to small, easy prey. Some of the bone fragments in the boluses come from animals that were themselves substantial. Other arthrodires, primitive sharks, animals that in most ecosystems would themselves be considered significant predators. Dunkleosteus was not hunting at the bottom of the food web. It was consuming other predators, actively and regularly. This makes ecological sense. An animal of its size required an enormous caloric input to sustain itself. Small fish consumed one at a time would simply not have provided sufficient energy return per hunting effort. The optimal prey for Dunkleosteus was large, energy-dense, and unfortunately for them, also present in the same waters. Other large placoderms, large primitive sharks, anything with sufficient mass to justify the energy expenditure of the hunt. And here, a detail emerges from the fossil record that shifts our picture of this animal from powerful to something more unsettling. There is evidence, in the bone fragments recovered from Dunkleosteus boluses, of other Dunkleosteus. Fragments that, based on the thickness and structure of the bone, appear to come from individuals of the same species. Consumed, digested, expelled. Cannibalism is not rare in the animal kingdom. Modern sharks practice it. Large predatory fish practice it. The largest and most dominant individuals consume smaller or weaker members of their own species when other prey is scarce, or simply when opportunity presents itself. There is nothing morally significant about this. It is pure energetics, pure survival logic. But it tells us something about the competitive intensity of the world Dunkleosteus inhabited. Even at the top of the food chain, there was no safety. Even among the most powerful animals in the Devonian Sea, size and armor were not guarantees. The hierarchy was enforced by force, continuously, without mercy or exception. This was not a gentle world. This was not a world where the powerful rested easy. This was an ocean where even the apex predator had to remain, always, a predator, because the moment it became anything less, something would take advantage of the fact. And somewhere in those same dark waters, the first tentative experiments were underway that would eventually lead, after hundreds of millions of years of evolution, to something new, something that would outlast Dunkleosteus by an almost incomprehensible margin. The first fish were beginning to develop the anatomical structures that would, eventually, become limbs. The ocean that belonged to Dunkleosteus was, without knowing it, already preparing for the world that would replace it. Let us step back from the jaw for a moment, from the bone blades and the bite force and the boluses full of crushed competitors. Let us step back and ask a different question, one that paleontologists spent decades arguing about, and one that, even now, does not have a completely settled answer. How did Dunkleosteus move? Because the way an apex predator moves through its environment tells you almost everything about how it lived. A pursuit predator needs sustained speed and endurance. An ambush predator needs explosive acceleration and precise control. A wide-ranging territorial animal needs efficient cruising locomotion that can cover large distances without excessive energy expenditure. The locomotion strategy is not separate from the hunting strategy. It is the hunting strategy, expressed in bone and muscle and fluid dynamics. And for Dunkleosteus, reconstructing locomotion from the fossil record presents a unique and genuinely difficult challenge. We have the front half. We do not have the back half. This is the same problem that haunts every aspect of Dunkleosteus biology, returning again in the context of movement. The thoracic shield, the skull, the jaw, all of these are preserved in multiple specimens with enough detail to support serious biomechanical analysis. But the tail, the paired fins, the entire posterior musculature that actually drives the animal through the water, almost none of this has been found. It is gone, consumed by time and bacteria and the slow chemistry of decomposition before the sediment could lock it in place. So researchers do what paleontologists always do when the direct evidence is missing. They look sideways. They look at relatives. Among the more completely preserved placoderms, smaller arthrodires and other placoderm groups where the entire body is sometimes fossilized, certain patterns emerge. Many placoderms appear to have had a body shape somewhere between that of a modern shark and a modern ray, broad across the front, tapering toward the tail, with paired pectoral fins positioned relatively far forward on the body. The tail itself, in well-preserved specimens of related species, shows a structure called heterocercal. The spine curves upward into the upper lobe of the tail, which is larger than the lower lobe, in a configuration that generates both thrust and a downward pitching force on the rear of the body. This tail design, combined with the heavy armored front end, has led some researchers to propose that Dunkleosteus was not a fast open-water cruiser. The mass distribution, enormous weight concentrated at the front in the thoracic shield, relatively lighter musculature at the rear, would have created trim problems in open water. Too much weight at the bow, in nautical terms. An animal built this way would require constant active fin adjustment to maintain horizontal attitude, burning energy simply to stay level. One hypothesis, developed in part through computer fluid dynamics modeling, suggests that Dunkleosteus may have been primarily a reef-associated or bottom-associated predator. An animal that spent much of its time hovering near the seafloor or among shallow reef structures, using the bottom itself to help manage its trim, and launching upward or forward in short explosive bursts when prey came within range. This model fits with the ambush predator interpretation of the suction jaw mechanics. A predator that does not need to chase its prey over long distances does not need sustained swimming efficiency. It needs acceleration. It needs the ability to go from stillness to full speed in the shortest possible time and distance. But this interpretation has critics. Other researchers point to the muscle attachment scars visible on the thoracic shield and argue that the musculature powering the tail was substantial, more substantial than a slow-moving, bottom-lurking ambush predator would require. The cross-sectional area of muscle implied by those attachment points suggests an animal capable of generating real power, real speed, over at least moderate distances. Not a pursuit predator in the sense of a tuna or a mako shark, but not a sluggish bottom dweller either. A study published in 2017 using three-dimensional modeling of Dunkleosteus body proportions scaled from thoracic shield dimensions suggested that the animal may have been closer in its swimming dynamics to a large modern grouper than to either a shark or a ray. A powerful, relatively maneuverable predator capable of short bursts of speed, comfortable in complex shallow water environments, using both active pursuit over short distances and ambush tactics depending on prey type and local conditions. The honest answer is that we are still guessing. Informed guessing, constrained by physics and comparative anatomy, and an ever-improving set of computational tools. But guessing nonetheless. What we are more confident about is the sensory system. Modern research on placoderm cranial anatomy has revealed a picture of the sensory world of Dunkleosteus that is considerably more sophisticated than early reconstructions suggested. For a long time, the armored head of placoderms was assumed to limit sensory input. All that bone, the reasoning went, must have insulated the animal from subtle environmental signals. The image was of a blunt, heavily armored brute, relying on vision and little else. The image was wrong. CT scanning of placoderm skulls, including arthrodire specimens closely related to Dunkleosteus, has revealed extensive canal systems within the dermal bone, channels running through the armor in complex branching patterns, connecting to the surface through small pores. These canal systems are now interpreted as the housing for the lateral line system, the network of mechanoreceptive cells that allow fish to detect pressure waves and water movement at a distance. The lateral line is not a visual system. It does not depend on light. It detects the physical displacement of water caused by the movement of other animals, the bow wave of an approaching fish, the turbulence left in the wake of a fleeing prey item, the subtle pressure differential that indicates something large is nearby and moving. In modern sharks, the lateral line system is extraordinarily sensitive. A shark can detect the tail beats of a fish at distances of several hundred meters in the right conditions. There is no reason to believe the lateral line system of Dunkleosteus was less capable. In fact, given the canal density visible in well-preserved arthrodire skull material, some researchers have argued it may have been more extensive than in most modern sharks, potentially providing extremely precise three-dimensional mapping of the animal's immediate environment, regardless of light conditions.
This matters because the Cleveland Sea, like many stratified ancient oceans, was not necessarily a well-lit environment throughout its depth range. The anoxic lower layers were, by definition, cut off from productive photosynthesis and likely murky with organic matter. Even in the oxygenated upper layers, turbidity from algal blooms, river outflow, and resuspended sediment could dramatically reduce visibility. A predator that could hunt effectively in low visibility or in complete darkness would have had an enormous advantage over prey species dependent on vision for their own escape responses. Add to this the evidence for electroreception. Electroreception, the ability to detect the weak electrical fields generated by the muscle activity and nerve firing of other animals, is present in several groups of modern fish, most famously sharks, which use their ampullae of Lorenzini to detect the bioelectric fields of hidden prey at close range. In living sharks,
Electroreception allows the animal to locate a flatfish buried under several centimeters of sand, detecting the electrical signature of the fish's heartbeat and gill movements. The presence of electroreceptive structures in placoderms is less well established than the lateral line evidence, and the question of whether Dunkleosteus specifically possessed fully functional electroreception is still debated. But several closely related arthrodire species show cranial anatomy consistent with electroreceptive pit organs, and the evolutionary context, electroreception being an ancient vertebrate trait present in many early fish lineages, makes it plausible, though not proven, that Dunkleosteus had some version of this system. If it did, then the sensory picture of this animal becomes formidable in a new way. An apex predator that can detect movement at distance through pressure wave sensing, that can locate electrically active prey in complete darkness or zero visibility, that has forward-facing eyes providing binocular overlap for precise close-range targeting, that has a jaw capable of opening faster than a human can blink and generating bite forces sufficient to shear through bone, that is encased in armor that distributes and absorbs impact from any retaliatory strike. This is not a simple brute. This is a supremely sophisticated sensory and mechanical system, refined over tens of millions of years of evolution in a competitive ocean, optimized for the single purpose of locating, approaching, and destroying prey with maximum efficiency and minimum risk. Now ask yourself, what does an animal like this do to an ecosystem? The answer, according to modern ecological theory, is profound. Apex predators do not simply sit at the top of a food web and remove individuals from the populations below them. They reshape behavior. They create what ecologists call a landscape of fear, a set of spatial and temporal patterns in the behavior of prey animals that are driven not by direct predation, but by the constant risk of predation. Prey animals change where they feed, when they feed, how long they stay in any one location, how they structure their social groups, all in response to the presence of an apex predator, even when that predator is not actively hunting. In modern marine ecosystems, the presence of sharks changes the distribution and behavior of their prey species across entire ocean basins. Reef fish alter their feeding patterns. Mid-water fish change their depth preferences. The effects cascade downward through the food web in ways that are measurable and profound, a phenomenon called a trophic cascade, where the removal or addition of an apex predator reverberates through every level of the ecosystem below it. There is no reason to believe the Devonian was different. The presence of Dunkleosteus in the Cleveland Sea and its relatives in equivalent late Devonian seas around the world would have imposed a landscape of fear on every other animal in those waters. Cladoceloshi, that early shark, presumably altered its behavior around Dunkleosteus. The smaller placoderms that shared those waters would have had behavioral adaptations, depth preferences, timing of feeding activity, schooling behaviors, shaped by millions of years of selection pressure from a predator that could kill them with a single bite. The armor that many Devonian fish carried, including the dermal bone plating seen in various placoderm groups far smaller than Dunkleosteus, may itself be partly a response to this predation pressure. Not all of it, certainly. Armor predates Dunkleosteus by tens of millions of years, but the particular density and distribution of armoring in late Devonian fish communities may reflect an arms race partly driven by the presence of an apex predator with the jaw force to make soft tissue completely indefensible. Dunkleosteus did not merely live in its ecosystem, it built it, it shaped it. It was the central organizing force around which every other behavioral and evolutionary strategy in the late Devonian Sea was arranged. And then something happened. Something so large, so catastrophic, so total in its destruction of the world, that Dunkleosteus had ruled for 40 million years, that when the dust, or rather the sediment, finally settled, nothing in the ocean would ever quite look the same again. The late Devonian mass extinction was not a single event. It was a series of pulses, a prolonged crisis that unfolded over millions of years, marked by at least two major extinction peaks: the Kelvasser event at the Frasnian-Fominian boundary, approximately 372 million years ago, and the Hagenberg event at the very end of the Devonian period, approximately 359 million years ago.
Between them, these two pulses erased more than 70% of all species on Earth. And the ocean that emerged from the other side was barely recognizable as the same body of water. But the story of how Dunkleosteus met its end, and what that ending meant for the future of life on this planet, is a story with more complexity, more scientific debate, and more genuine unresolved mystery than most accounts of extinction events allow. That story begins not with a catastrophe, but with a slow creeping change in the chemistry of the ancient sea. The ocean does not die all at once. It suffocates, slowly, in stages, over thousands of years that compress, in the geological record, into layers of dark sediment so thin you could cover them with your thumb. But thin as they are, those layers tell a story of environmental collapse so complete, so systematic, that geologists who first identified them in the 1970s and 80s struggled to find a modern analog. There was nothing in the current world quite like what those sediments described. You had to go back, further back, to a world operating under different rules, with different chemistry, with different vulnerabilities than the ocean we know today. The first pulse of the late Devonian extinction is called the Kelvasser event. It takes its name from a black shale horizon exposed in the Rhenish Massif of Germany, a thin, dark band of anoxic sediment that marks a sudden, catastrophic drop in ocean oxygenation approximately 372 million years ago, at the boundary between the Frasnian and Fominian stages of the late Devonian. The same horizon, or its equivalent, has been identified on every continent: in Morocco, in North America, in China, in Australia. It is a global signal preserved in rock of something that happened to the entire ocean simultaneously. The water ran out of oxygen. Not everywhere, not at every depth, but in the shallow, warm, productive epicontinental seas that covered much of the world's continental shelves, precisely the waters where the most diverse and abundant marine life was concentrated, oxygen levels crashed. The warm, stratified water column, already prone to anoxia in its deeper layers, lost its vertical circulation. The boundary between oxygenated surface water and anoxic deep water moved upward. Dead zones expanded from the bottom, rising through the water column like a slow tide of chemical darkness. Marine organisms that needed oxygen, which is to say nearly all of them, were squeezed into an ever-narrowing band of habitable water near the surface. And as that band compressed, competition for space and resources within it intensified catastrophically. Animals that could tolerate low oxygen, certain invertebrates, microbial communities, survived and expanded. Animals that could not, reef-building organisms, many groups of fish, the complex communities of the carbonate platforms, collapsed. The reef systems of the Devonian were among the most elaborate the world had yet seen. Stromatoporoid reefs, built primarily by sponge-like organisms called stromatoporoids, with contributions from tabulate and rugose corals, covered vast areas of the shallow continental shelves. These reefs were the ecological anchors of Devonian marine communities. They provided structure, habitat, feeding grounds, spawning sites.
the nurseries where juvenile fish of dozens of species spent their early lives. The Kellwasser event did not merely damage these reefs, it ended them. Stromatoporoid reefs disappeared from the fossil record at the Kellwasser horizon and did not return, not in any recognizable form, for millions of years. The reef-building ecosystem that had structured Devonian marine life for tens of millions of years simply ceased to exist. What replaced it in the immediate aftermath was not another reef system, but open, unstructured water.
A sea without architecture. A sea where the complex spatial organization of the Devonian marine community, the layered, niche-partitioned, reef-associated ecosystem that had supported the extraordinary diversity of the age of fishes, was gone. What caused the oxygen crash? This is the question at the center of the late Devonian extinction debate, and the honest answer, even today, is that we do not have a single agreed-upon explanation. What we have is a set of competing hypotheses, each supported by different lines of evidence, none of them sufficient on its own to fully explain the pattern of extinctions. The leading hypothesis, supported by substantial geochemical evidence, involves the forests. Remember those trees, the first forests spreading across the Devonian continents. Archaeopteryx and its relatives pushing inland, developing deep root systems, breaking down rock at rates the planet had never experienced before. The forests were not just changing the land, they were changing the ocean. As tree roots broke down continental rock, vast quantities of nutrients, phosphorus in particular, were released into rivers and carried downstream to the sea. Phosphorus is the key limiting nutrient in most marine ecosystems. When it arrives in large quantities, it triggers explosive growth of phytoplankton and algae in the surface waters. The algae bloom. They die. They sink. Bacteria in the water column consume the dead organic matter, and in doing so, consume oxygen. As the volume of sinking organic material increases, driven by the nutrient influx from the forests, the bacterial oxygen demand in the water column grows. Oxygen levels drop. Dead zones expand. The technical term for this process is eutrophication. It is not a theoretical concern. It happens today in coastal waters receiving agricultural runoff rich in phosphorus and nitrogen. The dead zone in the Gulf of Mexico, an area of severely depleted oxygen caused by nutrient runoff from Midwestern farmland, is a small-scale modern example of the same basic mechanism. In the Devonian, the nutrient source was not farms, but an entire planet's worth of newly forested continents, releasing minerals at a scale and rate that the ocean had no precedent for and no buffering capacity to absorb. This hypothesis, sometimes called the forest and weathering hypothesis, is compelling. It connects two of the most dramatic events of the Devonian period, the rise of forests on land and the collapse of marine ecosystems in the sea, through a coherent geochemical mechanism. It explains the timing. It explains the global nature of the signal. And it is supported by carbon and sulfur isotope data from Kellwasser black shales that are consistent with massive organic carbon burial driven by algal blooms and anoxic conditions. But it is not the only explanation on the table. An alternative hypothesis, supported by evidence from oxygen isotope analysis of fossil material, proposes that the Kellwasser event was at least partly driven by global cooling, a rapid drop in sea surface temperatures caused by the drawdown of atmospheric carbon dioxide. The forests in this model are again implicated, but through a different mechanism. Trees consume carbon dioxide during photosynthesis and lock it into organic matter. The first global forests represented an enormous new carbon sink,
pulling carbon dioxide from the atmosphere at rates the planet had not experienced before. Reduced atmospheric carbon dioxide means a weakened greenhouse effect. A weakened greenhouse effect means cooling. Cool surface waters circulate more effectively than warm ones. Better circulation means better mixing of the water column. Better mixing brings oxygen-rich surface water down and nutrient-rich deep water up.
In some models, this increased upwelling could itself drive the eutrophication and anoxia observed in the Kellwasser black shales. In other models, the cooling itself was the primary killer. A rapid temperature drop exceeding the thermal tolerance of the warm-adapted Devonian marine fauna. And then there is a third hypothesis, one that for many years was considered fringe but has gained increasing traction as additional evidence has accumulated. Extraterrestrial impact. The evidence for a bolide impact at or near the Frasnian-Fominian boundary is not as clear or as compelling as the evidence for the end Cretaceous impact that killed the non-avian dinosaurs. There is no global iridium anomaly of the kind found at the Cretaceous-Paleogene boundary. There is no confirmed impact crater of the right age and size. But there are anomalies. Elevated concentrations of certain elements, including iridium and other platinum group metals, at some Kellwasser horizon sites. Shocked quartz grains, tiny crystals bearing the distinctive deformation features caused by the extreme pressure of an impact, reported from late Devonian sediments in several locations. The impact hypothesis remains contested. Most researchers in the field consider it insufficient as a primary explanation for the Kellwasser event, even if some extraterrestrial input cannot be ruled out. But it illustrates something important about where the science stands. The late Devonian extinction is not a solved problem. It is an active research frontier, with new data arriving regularly and the consensus position shifting, sometimes significantly, with each major new study. What is not in dispute is the scale of what happened. The Kellwasser event eliminated approximately 40% of all marine genera.
Entire groups of organisms, brachiopod families that had existed for over 100 million years, trilobite lineages that had been a constant presence in marine ecosystems since the Cambrian, the reef-building stromatoporoids, were gone. The fish diversity of the Devonian seas, which had been extraordinary, was slashed. Dunkleosteus survived the Kellwasser event. This is important to understand. The first major pulse of the late Devonian extinction did not kill off the placoderms. Dunkleosteus terrelli and its relatives appear in Fominian age deposits, sediments laid down after the Kellwasser horizon, indicating that the lineage persisted through the first crisis. The world it inhabited was diminished. The reef ecosystems were gone. The diversity of its prey was reduced. But the animal itself, and the placoderm group more broadly, endured. For a while. Because approximately 13 million years after the Kellwasser event, at the very end of the Devonian period, the second pulse arrived. The Hagenberg event. And the Hagenberg event was different in character from the Kellwasser. Sharper, more sudden, more total in its impact on specific groups. Where the Kellwasser was a prolonged suffocation, a slow chemical crisis playing out over thousands of years, the Hagenberg appears in the geological record as something more abrupt, a crisis that, in geological terms, was almost instantaneous. The Hagenberg horizon is marked by a dramatic negative excursion in carbon isotope ratios, a geochemical signal indicating a massive and sudden disruption to the global carbon cycle. It is associated with evidence for rapid sea level fall, a drop in ocean levels significant enough to expose previously submerged continental shelves, destroying the shallow marine habitats that had already been stressed by the Kellwasser event. It is associated with renewed and even more intense ocean anoxia. And in some sections, it is associated with evidence for glaciation, ice formation at high latitudes, in a world that had been largely ice-free for tens of millions of years. The combination of rapid sea level fall, renewed anoxia, and possible glaciation hit the already weakened Devonian marine fauna with the force of a second hammer blow falling on a structure already cracked by the first. Among the victims of the Hindenburg event were the placoderms. All of them. Every single species, every genus, every family. The entire class Placodermi, the armored fish that had dominated the world's oceans for over 100 million years, that had produced the first vertebrate apex predators, that had driven the evolution of the jaw and the paired fin and body plans that would influence every vertebrate that came after them. Gone. Not reduced, not diminished, gone. The placoderms did not leave descendants. There is no modern lineage that traces its ancestry back through the placoderms. The sharks that survived the Hindenburg event and went on to populate the post-Devonian Ocean are not descended from placoderms. The bony fish that survived and eventually gave rise to every ray-finned fish alive today, from goldfish to tuna to the coelacanth lurking in deep water off the coast of Africa, are not descended from placoderms. Even we, the tetrapods, the land-walking vertebrates that emerged from the water during the Devonian and eventually produced every amphibian, reptile, bird, and mammal on Earth, we are not descended from placoderms. The placoderms were a dead end. A successful, dominant, extraordinarily diverse dead end, but a dead end nonetheless. And Dunkleosteus terrelli, the apex of that lineage, the largest and most powerful animal the placoderm experiment ever produced, went with them. The ocean it had ruled for 40 million years closed over the memory of it. The sediment accumulated. The dark shale compressed around the skull plates and jaw sections and thoracic shields of the last individuals, preserving them in the chemical stillness of the anoxic seafloor. And for 359 million years, there was silence. Until a quarry worker in Ohio picked up a piece of bone and wondered what he was holding. Here is the question that keeps paleontologists awake. Not the question of what killed the placoderms. We have hypotheses for that. Competing, unresolved, partially supported hypotheses, but hypotheses nonetheless. The question that is harder to answer, the question that cuts deeper into the foundations of how we think about evolution and survival and the logic of natural selection, is this. Why did the sharks survive? Because they did. The sharks, battered and diminished by the Hindenburg event, reduced in diversity to a handful of lineages, made it through. The early bony fish made it through. The first tetrapods, those extraordinary transitional animals in the process of hauling themselves out of the water and onto land, made it through. Life did not end at the Devonian boundary. It contracted severely and traumatically, and then it continued. But the placoderms did not. And the contrast between the placoderms and the sharks is not a contrast between a weak group and a strong one. It is not a story of superior design triumphing over inferior design. The placoderms had been successful for over 100 million years. They had survived previous environmental crises. They had diversified into virtually every marine ecological niche available to vertebrates. They had produced animals ranging from tiny bottom-dwelling filter feeders to the largest apex predators the oceans had yet seen. By any reasonable measure, the placoderms were one of the most successful vertebrate groups in the history of life on Earth. And yet, the sharks survived, and the placoderms did not.
So what exactly was the difference? Several hypotheses have been proposed, and the scientific literature on this question is genuinely divided. The first and perhaps most intuitive explanation involves reproductive strategy. Sharks reproduce slowly. They have long gestation periods, produce relatively few offspring, and invest significantly in each individual young. This is called a K-selected reproductive strategy, one that emphasizes quality over quantity, producing offspring with high individual survival probability rather than flooding the environment with vast numbers of low-investment young. It is, in general, a strategy associated with stable environments where resources are predictable and competition is the primary selective pressure. During an extinction event, this strategy might seem like a liability. Fewer offspring means slower population recovery after a crash. A species that produces 2 or 3 pups per year recovers from population collapse far more slowly than one that produces thousands of eggs. But there is a compensating factor. Sharks by the late Devonian had already developed a reproductive innovation that may have been critical to their survival. Several lines of evidence, including the discovery of articulated juvenile shark specimens preserved in adult body cavities, and the presence in some Devonian shark specimens of structures interpreted as claspers, the intromittent organs used for internal fertilization, suggest that some late Devonian sharks were already practicing internal fertilization and possibly live birth or ovoviviparity. Instead of releasing eggs into open water where they were vulnerable to predation, environmental fluctuation, and the chemical instability of an ocean in crisis, they retained developing young within the body. Internal fertilization and live birth are extraordinarily effective buffers against environmental instability. The developing offspring is not exposed to external conditions until it has reached a size and developmental stage where it can actively respond to its environment. In a world where the chemistry of the ocean was fluctuating catastrophically, where oxygen levels were crashing, temperatures were swinging, sea levels were dropping, carrying your young inside you, protected from all of that, would have been an enormous advantage. The placoderms also showed evidence of internal fertilization. This is actually one of the most remarkable recent discoveries in vertebrate paleontology. A 2008 study published in Nature, led by John Long of Museum Victoria in Australia, described a spectacularly preserved arthrodire placoderm specimen from the Gogo Formation of Western Australia, containing within its body cavity the mineralized remains of an embryo, complete with an umbilical cord-like structure. This animal, named Moderpiscus attenboroi, in honor of Sir David Attenborough, pushed the origin of live birth invertebrates back by hundreds of millions of years. Live birth did not begin with mammals or even with reptiles. It was already present in the Devonian, an armored fish, 380 million years ago. So both sharks and at least some placoderms had internal fertilization. The reproductive strategy alone cannot explain the differential survival. The second hypothesis focuses on skeletal composition. Sharks have cartilaginous skeletons, not bone, cartilage. This is often presented in popular accounts as a primitive feature, a sign that sharks are somehow less evolved than bony fish. This is wrong. Cartilage is not simply undeveloped bone. It is a different tissue, with different mechanical properties, different metabolic costs, and different developmental biology. And one critical difference, in the context of an environmental crisis, is that cartilage requires significantly less calcium to build and maintain than bone does. In a world where ocean chemistry is disrupted, where carbonate saturation is dropping, where the availability of dissolved minerals is fluctuating, The metabolic cost of building and maintaining a bony skeleton increases. Organisms that are heavily invested in calcified structures, thick bone, dense armor, heavily mineralized scales, face a higher metabolic burden in maintaining those structures when the chemical environment is stressed. Organisms with reduced calcification requirements have a lower baseline metabolic cost and are therefore more resilient to resource fluctuations. The placoderms were among the most heavily armored vertebrates that ever existed. The thoracic shield of Dunkleosteus alone represented an enormous investment in mineralized tissue. Maintaining that armor, replacing damaged plates, supporting its weight, building it in growing individuals, required sustained access to minerals and energy. In an ocean where productivity was crashing, where the nutrient availability was swinging wildly, where the very chemistry of the water was in upheaval, that investment may have become a fatal liability. The armor that had made Dunkleosteus invulnerable, the evolutionary innovation that had allowed the placoderms to dominate for over 100 million years, may have become, in the context of the Hindenburg crisis, too expensive to maintain. This is one of the most profound lessons the placoderms have to teach us about evolution. Adaptations are not universally beneficial. They are beneficial in the specific environmental context that produced them. Change the context dramatically enough, and the adaptation that guaranteed survival in the old world becomes the vulnerability that ensures extinction in the new one. The armor was not a mistake. It was a perfect solution to the selective pressures of the Devonian Ocean. And the Devonian Ocean was ending.
A third hypothesis, more recent in its development and still actively debated, concerns ecological specialization. The placoderms, by the late Devonian, had diversified enormously. But much of that diversification had produced highly specialized forms, tightly adapted to specific ecological roles in specific environments. The reef-associated species were adapted to reef environments. The shallow water predators were adapted to shallow water conditions. The deep shelf filter feeders were adapted to deep shelf conditions. This specialization made them extraordinarily efficient in the stable, productive Devonian seas. It made them extraordinarily vulnerable when those seas changed. The Hindenburg event did not just reduce the productivity of the ocean, it reorganized it. The shallow epicontinental seas that had covered the continental shelves, the warm, productive, reef-associated environments that had been the heart of Devonian marine diversity, were dramatically reduced by the sea level fall. The habitats that most placoderm species were specialized for simply shrank, in some cases to near non-existence. The early sharks, by contrast, appear to have been somewhat less specialized in their habitat requirements. Cartilaginous fish are found in a wider range of depositional environments in the late Devonian. Open ocean settings, deeper water, environments less tied to the shallow reef shelf systems that the placoderms dominated. When the shallow seas retreated, the sharks had somewhere else to go. The placoderms, in many cases, did not. There is one more factor that deserves mention, one that is speculative but intriguing. Several researchers have noted that the extinction selectivity of the Hindenburg event was not random. It did not simply erase a random sample of Devonian marine diversity. It was disproportionately lethal to large-bodied, heavily armored, reef-associated animals, precisely the characteristics that define the most successful placoderms, including Dunkleosteus. Small-bodied animals survived at higher rates. Less specialized animals survived at higher rates. Animals with broader habitat tolerances survived at higher rates. Dunkleosteus was large, heavily armored, associated with shallow, productive coastal seas. It ticked every box of the extinction vulnerability profile. But here is the detail that makes this more than simply a story of the wrong animal in the wrong place at the wrong time. Here is where the science becomes genuinely unsettling in its implications. The characteristics that made Dunkleosteus most vulnerable to the Hagenberg event, the large body size, the heavy armor, the specialization for productive coastal seas, were not weaknesses. They were the product of 40 million years of successful adaptation. They were the traits that natural selection had favored consistently and powerfully throughout the entire history of the placoderm lineage. They were the reasons Dunkleosteus had been the apex predator of the Devonian Ocean, the reasons it had survived and thrived while other, less specialized, less heavily invested animals had remained in the ecological background. Evolution had built the perfect Devonian predator. And then the Devonian ended. There is no foresight in evolution. There is no long-term planning. Natural selection operates on the present environment, rewarding traits that confer survival advantages now, with no consideration of whether those same traits will remain advantageous when conditions change. Every adaptation is a bet that the environment that produced the selection pressure will continue to exist long enough for the adaptation to pay off. Most of the time, the bet is good.
The environment is stable enough or changes slowly enough that adaptations remain relevant across many generations. But sometimes, the environment changes faster than evolution can respond. Sometimes the bet comes due all at once, called in by a catastrophic reorganization of the world, and the animal that was perfectly adapted to the old world finds itself precisely, specifically, fatally unsuited to the new one. This is not a comfortable lesson. It has implications that extend well beyond the Devonian. Every organism alive today is perfectly adapted to the current world, or rather, to the world that existed during its recent evolutionary history, which we assume closely resembles the current world. The specializations that make modern predators effective, that make modern ecosystems functional, that make the current biosphere the extraordinarily productive system it is, all of these are bets. Bets that the conditions producing them will persist. The placoderms made the same bet for 100 million years and won, consistently and spectacularly, until the moment they lost everything. But the story does not end there, because the same extinction event that destroyed the placoderms opened a door. The ecological collapse of the Hagenberg event did not leave the ocean empty. It left it reorganized, simplified, stripped of the complex, niche-partitioned community structure of the Devonian, and reset to something more open, more dynamic, more available for new evolutionary experiments. And into that newly available ocean, in the early Carboniferous period immediately following the Devonian boundary, the survivors expanded. The sharks diversified explosively. The bony fish, the ray-finned actinopterygians that had been a relatively minor component of Devonian fish diversity, began their own extraordinary radiation, eventually producing a diversity of forms that would come to include the vast majority of all fish species alive today. The tetrapods, already ashore, continued their conquest of the land. The world that emerged from the late Devonian extinction was not a lesser version of the Devonian world. It was a fundamentally different world, a world organized around different body plans, different ecological strategies, different evolutionary possibilities. A world in which the placoderms had no place, because the placoderms were gone, but in which the innovations the placoderms had developed and refined over their 100 million years of existence lived on. Not in their descendants, they had none, but in the logic of the ocean itself, in the ecological template of apex predation they had established, in the evolutionary pressure they had exerted on every other lineage that shared their world.
The sharks that inherited the Devonian Ocean did not inherit it from the placoderms, but they inherited a world that the placoderms had helped shape. An ocean whose ecological structure, whose prey communities, whose depth distributions and habitat complexities had all been molded in part by tens of millions of years of placoderm dominance. Dunkleosteus did not have children, but it had consequences, and those consequences echo in ways both direct and indirect all the way to the present day. The final chapter of the Dunkleosteus story is not written in rock. It is written in what came after, in what its world produced, and what its extinction made possible, and what questions remain open in the shale of Ohio and Morocco and China and everywhere else this extraordinary animal left its mark on the planet. Those questions are waiting in the last layer of sediment, and we are only beginning to read them. Pick up a piece of Cleveland shale. It fits in your hand. It is dark, almost black, dense and smooth, with a faint organic smell that is not quite earth and not quite sea, but something older than either. If you split it along its natural planes, it opens like a book. And sometimes, when it opens, there is something inside. A scale, a fin ray, a fragment of bone plate with the surface texture still visible, the tiny ridges and tubercles that covered the outer surface of a Dunkleosteus armor plate, preserved in such detail that you can run your fingertip across them and feel the texture of an animal that died 359 million years ago. This is what paleontology actually feels like. Not the dramatic excavation scenes of documentary films, not the grand museum reconstructions under soaring ceilings, but this, a piece of rock in your hand, and the slow, careful realization that you are touching something real. There is a skull in the Cleveland Museum of Natural History that has been studied by more researchers, measured more carefully, scanned more thoroughly, and argued about more extensively than perhaps any other single Devonian vertebrate specimen in the world. It is not complete. It is never complete. That is the fundamental condition of Dunkleosteus research, the absence at the center of everything. But it is extraordinarily well preserved. The bone surface is intact. The joint surfaces between individual armor plates are clear. The orbital cavities, where the eyes sat, are present and measurable. The jaw, in multiple associated pieces, preserves the bone blade edges in enough detail to allow the kind of geometric analysis that Anderson and Westneat used to model the bite mechanics. Researchers have been returning to this specimen and to the broader Cleveland Museum collection for over a century, and each time they return, they bring new tools, new questions, new analytical techniques that extract information from the bone that previous generations of researchers had no way to access. In the early decades of Dunkleosteus research, the tools were simple: caliper measurements, anatomical description, comparison with other known placoderms by eye and intuition. The reconstructions produced from these methods were not wrong exactly. The fundamental anatomy was correctly identified early, but they were coarse. They captured the outline of the animal without resolving its details. Then came CT scanning, high-resolution computed tomography, the same basic technology used in medical imaging, applied to paleontological specimens, transformed the study of fossil skulls in the 1990s and 2000s. For the first time, researchers could look inside bone without cutting it. They could trace internal channels and cavities, map variations in bone density, identify structures that were completely invisible on the external surface. Applied to Dunkleosteus skull material, CT scanning revealed the canal systems that house the lateral line, internal structures within the armor plates that inform our understanding of the muscle attachment geometry, and details of the joint surfaces between skull plates that have been critical to refining the craniokinesis model. Work using CT data from Dunkleosteus specimens, combined with three-dimensional digital reconstruction and computational fluid dynamics modeling, is ongoing at multiple institutions. The biomechanical models published since Anderson and Westneat's 2006 paper have refined and, in some cases, significantly revised the earlier estimates. Jaw opening speed estimates have been revisited. The suction feeding hypothesis has been developed in greater detail, with some researchers arguing the suction component was more important than the cutting component in initial prey capture, with the bone blades serving primarily to process and consume prey already drawn into the mouth rather than to seize it from open water. This is a meaningful distinction. An animal that primarily uses suction to capture prey does not need to be fast in its approach. It needs to be close, patient, capable of maneuvering slowly into proximity with prey before triggering the jaw mechanism. This fits the ambush interpretation. It also fits the lateral line and electroreception picture. A predator that navigates by sensing the pressure fields and bioelectric signatures of nearby animals, closing in gradually, waiting for the moment when distance drops below the threshold where the suction becomes irresistible. A ghost in the water, silent and armored and ancient. Not the roaring, charging predator of popular imagination, but something more deliberate, more calculating in its mechanics. Something that did not need to be fast because it had made speed irrelevant. By the time its prey knew it was close enough to strike, it was already too late. But even as the functional biology of Dunkleosteus becomes clearer, other aspects of its biology remain genuinely mysterious. We do not know how it reproduced. The Mater piscis discovery demonstrated that internal fertilization and live birth existed in arthrodire placoderms by the late Devonian. But Mater piscis was a small, relatively delicate arthrodire from a different family. Whether the reproductive biology of large apex arthrodires like Dunkleosteus was similar, whether Dunkleosteus gave birth to live young or retained fertilized eggs internally or had some other strategy entirely, is unknown. No juvenile Dunkleosteus specimens have been identified with certainty. No reproductive structures have been preserved. The question of how this animal propagated itself, how many young it produced, how large they were at birth or hatching, how long they took to reach maturity, whether they received any parental investment, is completely open. This matters for understanding its ecology. An apex predator that produces few, large, well-developed young has a very different population dynamics profile than one that produces many small, vulnerable young. The time required to reach sexual maturity, the generation length, the population growth rate, all of these variables shape how a species responds to environmental stress, how quickly it can recover from population crashes, how resilient it is to the kind of sustained environmental crisis that the Hagenberg event represented. We also do not know how long individual Dunkleosteus lived. In modern large vertebrates, bone histology, the microscopic structure of bone tissue, including growth rings analogous to tree rings, can be used to estimate individual age and growth rate. Applied to dinosaur bone, this technique has produced detailed growth curves showing how quickly different species grew when they reached sexual maturity and approximately how long the largest known individuals had been alive. The results revolutionized our understanding of dinosaur biology, revealing growth rates far higher than anyone had expected and lifespans surprisingly comparable to large modern birds and reptiles. In principle, the same technique could be applied to Dunkleosteus dermal bone. In practice, Dermal bone grows differently from endochondral bone, the bone formed from a cartilaginous template that makes up most of the skeleton in bony vertebrates, and the growth ring interpretation is less straightforward. Some work has been done on placoderm bone histology, and the results suggest that at least some arthrodires had relatively rapid growth rates and indeterminate growth, continuing to grow throughout their lives, with the largest individuals simply being the oldest ones. But the data are limited, and the conclusions are tentative. We cannot say with any confidence how old the largest Dunkleosteus individuals were. We cannot say whether they grew quickly or slowly. We cannot say whether an 8-meter individual was 20 years old or 200 years old. We do not know with precision how many species of Dunkleosteus actually existed.
The taxonomy of the Dunkleosteus genus has been revised multiple times over the decades, and the question of which specimens represent distinct species versus geographic or age variants of existing species is unresolved. Different researchers, applying different species concepts and different morphological criteria, have recognized anywhere from 2 to over 10 species within the genus. The most widely cited current taxonomy recognizes around 10 species, ranging in size from relatively small to the giants of the Cleveland Shale. But this taxonomy is contested, and new specimens continue to complicate the picture. The species from Morocco deserve particular attention. Late Devonian placoderm material from the Anti-Atlas region of Morocco has produced Dunkleosteus specimens of remarkable quality and, in some cases, remarkable size. The Moroccan material has been critical in expanding our understanding of the geographic range of Dunkleosteus, demonstrating that this genus was not limited to the shallow seas of North America, but was distributed across much of the ancient Devonian Ocean, wherever the warm, shallow, productive conditions it preferred could be found. Some Moroccan specimens suggest individuals potentially larger than any yet found in Ohio, though the fragmentary nature of the material makes precise size estimates uncertain. Polish and Belgian deposits have also yielded Dunkleosteus material. So have deposits in North Africa more broadly, in what was the southern margin of Laurussia during the late Devonian. The picture that emerges is of an animal that was not merely a local apex predator in a single sea, but a widespread, successful genus distributed across much of the tropical and subtropical Devonian Ocean. A top predator at a genuinely global scale. This global distribution has implications for understanding the extinction. If Dunkleosteus was widespread across many basins and environments, then its complete disappearance at the end of the Devonian was not a local event, not the loss of one population in one stressed environment. It was a simultaneous global collapse. Every population in every ocean basin where the genus had established itself failed at approximately the same time. That speaks to the systemic, global nature of the Hagenberg event in a way that individual site records cannot. It was not enough to have a refuge somewhere else. There was no somewhere else. The crisis was everywhere, and it reached every population of every placoderm species on the planet with equal finality. And yet, and this is where the story takes a turn that most popular accounts of Dunkleosteus never reach, the placoderms are not entirely gone. Not in the literal sense. No placoderm lineage survived into the Carboniferous. The class Placodermi is genuinely, completely extinct. But recent work in vertebrate phylogenetics, the study of evolutionary relationships based on anatomical and genetic data, has produced results that have dramatically revised our understanding of where placoderms fit in the vertebrate family tree. For much of the 20th century, placoderms were considered an outgroup to all living jawed vertebrates, an ancient armored lineage that branched off from the vertebrate tree before the split between sharks and bony fish, flourished for a time, and then went extinct without contributing anything to the lineages that followed.
In this view, placoderms were an evolutionary experiment that simply ended, an impressive but ultimately irrelevant side branch. Recent phylogenetic analyses have challenged this picture.
A series of studies, including work published in Nature in 2013 by researchers including Martin Brazo and Matt Friedman, used new anatomical data from placoderm specimens, including detailed analysis of the jaw structure, the brain case, and the fin skeleton in well-preserved specimens, to propose that the placoderms may not be a natural group at all. In this view, what we call placoderms, the armored fish with the articulated skull and thoracic shield, may be a grade rather than a clade, a series of evolutionary stages leading toward the jawed vertebrates rather than a single unified lineage. Under some of these new phylogenetic hypotheses, the ancestors of the bony fish, and therefore ultimately the ancestors of every tetrapod, including ourselves, passed through a placoderm-like stage. The bony fish did not merely coexist with placoderms and then replace them. They emerged from among them. The armor may have been lost, the skull may have been reorganized, the jaw mechanics may have been rebuilt, but the fundamental vertebrate body plan that underlies every living jawed animal may trace its origins, at least in part, through the placoderm lineage. This is still contested. The phylogenetic position of placoderms relative to living vertebrate groups is one of the most actively debated questions in vertebrate paleontology. Different datasets, different analytical methods, different taxon samples produce different trees. The field has not reached consensus. But the possibility, even the possibility, that we are, in some deep evolutionary sense, modified placoderms is extraordinary. That the jaw mechanics of every predator alive today, every shark, every crocodile, every eagle, every human being who has ever bitten into an apple, are descended from the same fundamental vertebrate jaw innovation that Dunkleosteus carried to its most extreme and terrifying expression. That when Dunkleosteus opened its jaws in the shallow Devonian Sea, it was, in a sense, opening something that would never fully close. Now it is 359 million years later. The Cleveland Sea is gone. The rock that formed its floor has been uplifted, compressed, weathered, and in places eroded away entirely. What remains is exposed in cliffs and quarries and road cuts along the southern shore of Lake Erie, where occasionally, when the shale splits cleanly, the black surface of a bone plate catches the light. There are perhaps a few thousand reasonably significant Dunkleosteus specimens in collections around the world. Most are fragments, individual armor plates, isolated jaw sections, scattered pieces of a puzzle for which we have never found the box. The mounted reconstructions in the Cleveland Museum, the Field Museum in Chicago, the Natural History Museum in London, the Musée des Sciences Naturelles in Brussels, all of them are composites, assembled from multiple individuals, scientifically reviewed and revised as new specimens and new analytical techniques inform our understanding. None of them are complete. None of them ever will be. And this is, in its way, the most honest thing about Dunkleosteus. It is an animal we will never fully know. Not because the evidence is lacking. We have more and better evidence now than at any point in the history of the science, and the quality of that evidence is improving every year. But because the completeness we crave, the whole animal, the living behavior, the sound it made and the way it moved and the precise moment of its last breath, is beyond recovery. It lived and died in a world so ancient that the ocean floor that buried it has been subducted back into the mantle and replaced. What we have are the parts that were hardest to destroy, preserved by the specific accident of anoxic chemistry in a specific shallow sea,
recovered by the specific accident of a quarry worker in Ohio who happened to look at the right piece of rock on the right day. This is what extinction means, finally. Not just the death of individuals, everything dies. Not just the end of a species. Species end constantly, and the fossil record is the proof of that. Extinction means the permanent, irreversible closure of an evolutionary possibility. Every animal that goes extinct takes with it not just its own existence, but every variation, every adaptation, every form it might have become across all the millions of years of evolution it will never have. The placoderms, in their 100 million years, had explored one corner of the vast space of possible vertebrate body plans. The corner they explored was extraordinary. But when the Hangenberg event closed the door, it closed it on everything they had not yet become as well. That loss is real. It is not sentimental to recognize it. It is simply true. But here, at the end of the Dunkleosteus story, or rather at the end of what we currently know of it, because the story itself is still being written in laboratories and quarries and journal articles around the world, it is worth sitting for a moment with the scale of what this animal actually was. 380 million years ago, in a warm, shallow sea that would one day be Ohio, there was a predator. It was armored in bone. Its jaw was a biological machine of extraordinary precision and power. It had been shaped by evolution over tens of millions of years into something close to optimal. Not perfect, because nothing in evolution is perfect, but as close to optimal for its specific time and place as the blind process of natural selection can produce. It patrolled those waters for 40 million years. It shaped the evolution of every animal that shared its ocean. It imposed its logic, the logic of bone and blade and explosive jaw mechanics, on an entire ecosystem, bending the behavior and the anatomy and the survival strategies of every creature within its range. And then the world changed, and it was gone. And we, 359 million years later, are still finding its bones, still arguing about how big it really was, still debating how it moved, how it hunted, how it reproduced, how it died, and what exactly its relationship is to the vertebrate body plan that eventually produced, among many other things, us. There are new specimens being prepared right now in museum collections, new CT scans being processed, new phylogenetic datasets being assembled, new questions being asked of bones that have been sitting in drawers for 100 years, waiting for the right technology to make them speak. The shale is still full of Dunkleosteus. The quarries are still there. The dark layers of the Cleveland Formation still split open along their ancient plains, still occasionally revealing a surface of textured bone that has not been exposed to light since the Devonian sun went down for the last time over the shallow sea that is now Ohio. We are not done learning from this animal. We have barely started. And somewhere out there, in a cliff face not yet exposed, in a layer of shale not yet quarried, in a piece of rock sitting on a shelf that no one has yet thought to split open, there is a specimen that will change everything we think we know. The ocean kept its secrets for 359 million years. It is in no hurry to give them all up at once. There is a specimen that will change everything we think we know. The ocean kept its secrets for 359 million years. It is in no hurry to give them all up at once.